Most drug discovery tools have a hidden problem. They learn from the past. They are trained on databases of known drug targets, known binding sites, known molecules, and that means they can only recognize what they have already seen. What happens when the target is new? What happens when the binding site has never been drugged before? What happens when the protein comes from a pathogen nobody has studied, or a synthetic biology construct that has never existed in nature? These tools fail, not because the science is wrong, but because the assumption is wrong. They are pattern matchers pretending to be physics engines. The Ashibo method is different at the foundation. It does not use machine learning. It does not use training data. It does not know whether a protein is human or bacterial, familiar or novel, well-studied or completely unknown. It reads one thing only: three-dimensional atomic coordinates. From those coordinates, the engine computes a physical quantity called the local potency density field, the LPD field. This is a measure of where energy is concentrated in molecular space. Where the field peaks, the physics is telling you this is where a molecule wants to bind. No assumptions, no priors, no memory of what came before. Just the geometry of atoms in space and the physics that governs them. To understand why this matters, consider MDM2, one of the most important cancer targets known. It controls the tumor suppressor p53. When MDM2 is overactive, p53 is silenced and cancer cells survive. Drugs that block MDM2 can restore p53 function and kill tumor cells. We ran the Ashibo engine on MDM2 3 times. First, on an AlphaFold structure, the computationally predicted form of the protein. The groove where drugs bind was partially closed. The engine scored it 32 out of 100, very low druggability, a false negative by any clinical standard. But here is the key insight. The engine was not wrong. The coordinates it received genuinely had a closed groove. It reported exactly what the physics of those coordinates said. The problem was the input, not the engine. So we ran it again on the crystal structure of MDM2, captured with the drug Nutlin 3A bound, what is called a hollow structure. Now the groove was open, held open by the drug itself. The engine scored it 94 out of 100. And when we looked at which residues the engine identified, ILE99, MET98, LEU78, LEU81. These are the exact residues that crystallography has confirmed make contact with Nutlin 3A. The engine found the right atoms in the right location for the right physical reasons. No training data told it where to look. The field told it. We then ran it on BCL2, the target of venetoclax, one of the most successful cancer drugs of the last decade. The engine found the BH3 binding groove. It identified CYS158, GLY154, GLY155, MET157, LEU137, all confirmed venetoclax contacting residues. Again, no prior knowledge of venetoclax, no training on BCL2 structures, just atomic coordinates and physics. This is what coordinate native computation means. The engine is model agnostic, crystal structure, NMR ensemble, AlphaFold prediction, cryo-EM density, MD simulation snapshot. It processes all of them identically because they are all just atoms in space. It is species agnostic. Human proteins, bacterial proteins, viral proteins, plant enzymes, synthetic constructs, same physics, same engine, no retraining required, and it is assumption-free in the deepest sense. It will find a druggable site that has never been found before, in a protein that has never been studied before, because it is not asking whether this looks familiar, it is asking what the physics actually says. The competing tools ask, does this protein look like something we have seen before? The Ashibo engine asks, what does the physical field of this coordinate set actually look like? That is a more fundamental question. And for the targets that matter most, the novel ones, the resistant ones, the ones that have defeated every existing approach, it may be the only question that works. The Ashibo method, physics-based drug discovery, no training data, no assumptions, just the geometry of molecules and the laws that govern them. Character count, approximately 4,490.